Good morning. This video is an important video because it talks about transcription, mainly of RNA from DNA. And this video also fills the gap between part one, the video that was dedicated to part one, and the videos that were dedicated to to part two, because I found out that transcription wasn't explained in full detail. And because of this, and because we are now reviewing molecular biology, then it will be a kind of explication, but at the same time, it will include questions and answers about transcription. Let's first discuss the central dogma of molecular biology, which is from gene to protein. So, as you can see in this diagram, that a DNA molecule is a long molecule, and in interphase it gets duplicated. And here we can see uh, a non-duplicated DNA molecule, but still it is a double helix, meaning that when you see two strands that are helical, that are twisted over each other, this doesn't mean that it is a replicated DNA molecule. It's only one DNA molecule consisting of a double helix. What is a double helix? A double helix is two strands, two polynucleotide strands that are twisted around each other. So this is a non-duplicated DNA molecule that consists of a double helix or two strands of polynucleotides. But meanwhile, this molecule has specific segments, nucleotide segments, that are called the genes. So what is a gene? A gene is a part of the DNA molecule that is responsible for the formation of a particular polypeptide. And the polypeptide that is formed in such a case will be responsible for the trait that the gene determines. It will be responsible for the phenotype that is seen or produced when the gene is expressed. So when a sequence on DNA is translated into a polypeptide, and that polypeptide is a functional or structural protein, like a hormone, like an enzyme, like an antibody, or even a structural protein like tubulin, dimers, for example, or actin filaments, or motor proteins, then all this corresponds to a characteristic, whether this characteristic is a physical outer characteristic or an internal characteristic, like, for example, the formation of antigens on the surface of the red blood corpuscles, the development of internal organs. All these are traits and phenotypes, but they are inside the body. So traits and phenotypes can be outside or inside, can be visible to you or not. You sometimes see the internal organs, for example, when you have a surgery, medical doctors see them, but you can detect the presence of the organ through its function. So production of an enzyme by the liver, this is a phenotype. Production of a hormone by an endocrine gland, this is a phenotype. So how do we determine the length of the gene? The length of the gene is that part of the DNA molecule that is translated into a polypeptide that provides a trait or a phenotype. As you can see, the DNA molecule is very long, and therefore it consists of several genes. This is just a small segment of the DNA molecule. In the process of, or in this process of expressing a gene into a protein, and this arrow would represent which process. It would represent expression. We have two stages. We first have to transcribe DNA into messenger RNA, and then to translate messenger RNA into a protein. Translation has been explained or was explained explicitly in the videos that were sent to you, but it's only transcription that I'm focusing on. So what is transcription? Transcription is using a particular stretch of DNA or a particular segment of DNA to form a complementary strand of messenger RNA. So this complementary this strand you can see in purple is complementary to only that blue segment of DNA. And actually, what do we transcribe? We transcribe the gene. But in order for transcription to occur, you need to separate to unwind first the DNA molecule, because as you can see, it is helical. And then after unwinding the DNA molecule, you separate the two strands by breaking the hydrogen bonds in order to expose the nitrogenous base. That's why you separate the two strands. And as you can see, now we have those nitrogenous bases like adenine, cytosine, guanine, and thymine. They are exposed. What's the importance of exposing the nitrogenous bases? It's allowing the enzymes of transcription to have access to the nitrogenous bases and to read the code on DNA and to get the complementary nitrogenous bases to form a messenger RNA strand. Why is it necessary that the bases on messenger RNA are complementary to those on the template strand of DNA? Because as you know from the previous explanation and from the previous questions, that the strand that we use to transcribe is the template strand. And as you can see, this template strand will be read by the enzyme of transcription in the direction 3' to 5'. Why? What does this mean, read? That we read, or that the enzyme reads the template strand in the direction 3' to 5'. This simply means that the enzyme will move onto the template strand in the direction of the arrow that I've just put on the screen, and that is the direction 3' to 5'. Is it important for the enzyme to move in that direction? Yes, of course, it is important, because it is this direction that will allow the enzyme to build the new strand of messenger RNA in the direction 5' to 3'. So, what is then the enzyme that is responsible for the process of transcription? It is the RNA polymerase enzyme. So, what is the function of the RNA polymerase enzyme? It is the enzyme that is responsible for the actual building of the newly transcribed messenger RNA in the direction 5' to 3'. This is the only direction that polymerases can build new strands, whether of DNA in case of DNA polymerase or RNA in case of RNA polymerase. And at the end of the day, we have a template strand of DNA and a transcribed strand of messenger RNA. Every 3 consecutive strands of nitrogenous bases on the DNA template strand is called the triplet code. And we usually write, for example, AGT, but we write the direction 3' to 5'. So you can know that the enzyme will work in this direction. But every 3 consecutive strands on an RNA molecule are not called a triplet codon, but are called, a triplet code, but are called a codon. And every codon is responsible for one amino acid. Every codon is responsible for one amino acid. Moreover, there are some amino acids that are represented by more than one codon in the table of codons. What is translation? Translation is using a stretch of messenger RNA or a certain sequence of messenger RNA that corresponds, of course, to the original gene sequence in order to form a chain of amino acids, which is a polypeptide chain. Of course, when the polypeptide chain is formed, the sequence and the type first of amino acids and their sequence together in the chain should correspond to the sequence of codons on the messenger RNA molecule. So the triplet code, which is 3 consecutive nitrogenous bases on DNA, will be transcribed into a codon sequence in messenger RNA. But where does this process occur? It occurs inside the nucleus. In eukaryotes in particular, if you're talking about human beings, in eukaryotes in particular, the RNA that is freshly transcribed on the template strand of DNA is considered an immature RNA, meaning that it needs further processing in order to start functioning. So it's called a pre-RNA or an immature RNA. But before leaving the nucleus, I'm talking again about eukaryotes. Before leaving the nucleus, it has to be processed. It's just like, for example, immune cells in the thymus should get mature before leaving the thymus to act as mature lymphocytes. So it's the same. The immature RNA is processed in the nucleus, and afterward, it will leave the nucleus. How does it leave the nucleus? Through the nuclear pores. So if this is the nucleus of the cell, so a messenger RNA molecule that has been freshly transcribed will first be processed and then, and then will leave the nucleus to the cytosol, but of course will not leave outside the cell. Why does it leave to the cytosol? Because it is in the cytosol that the process of translation of RNA, of messenger RNA in particular, occurs. So why is it messenger RNA that is used in translation? Because messenger RNA is the only type of RNA that is a real delegate of DNA in the nucleus. It is the one whose sequence accurately represents the template strand of DNA. It's true that it's not a copy of it, but it is complementary. So it is meant that messenger RNA is complementary, because when you have, for example, CGA on the template strand of DNA, this is the template strand of DNA, and then you transcribe a complementary strand of messenger RNA, it would be like this. So this is messenger RNA. So when it goes to the cytosol and the enzymes start getting the complementary strands and build so when the enzymes start reading the codons on messenger RNA, they can deduce the triplet codes that were present on DNA and can get the corresponding amino acids. This is also very important in case of a particular group of enzymes that are called the reverse transcriptases in viruses, when the virus needs to convert its DNA, its RNA into a DNA. So it will, when it makes the complementary bases, the resultant strand would be an exact copy of the original DNA strand. So transcription is thus the process, as I've just said, by which the info in a DNA sequence is used to make a complementary sequence of RNA. And you have a good example here. And here, every 3 consecutive nitrogenous bases are called the triplet codes. So you have to get the complementary nitrogenous bases, taking into consideration, of course, that the complementary of A is U on messenger RNA. So once you see a stretch of nucleic acids where you have uracil, then it's definitely RNA and not DNA. So you have to, sometimes we get you this sequence and we ask you to get the complementary sequence, and in such a case, you have to respect the direction. So instead of, or in front of 5 prime, you put 3 prime, and in front of 3 prime, you put 5 prime. What is the enzyme that is responsible for the actual building of the messenger RNA strand? It is, of course, the RNA polymerase. Since it is a polymerase, then it makes a polymer. It makes a polymer from the ribonucleotides that are available in the nucleus. And as you can see, we have exactly the same structure that we saw in DNA replication. It looks like a bubble, but here it wouldn't be a replication bubble. It would be a transcription bubble, although we don't use this term frequently, but you need to separate the 2 strands first, and then the DNA polymerase starts building up the new strand. The messenger RNA strand that you can see, which is the red one, is complementary to the template strand of DNA. And as we said before, if you look at the bases, they will be exactly similar to the ones on the non-template strand, but with the difference of uracil instead of thymine. As you can also see, RNA polymerase always adds new nucleotides to the 3 prime end of the growing strand. And if we dive more deeply into the process of transcription, you will find that the RNA polymerase will also require a primase enzyme. It is that primase enzyme that builds up the first sequence of RNA, to which, or whose 3 prime end will be the place where RNA polymerase enzyme will start building the new strand of messenger RNA. And here it is important to mention something. When we talked about replication, we had the enzyme DNA polymerase. And when we talked about transcription, we had the enzyme RNA polymerase. And both are similar in the fact that they add to the 3 prime end. The new bases are added to the hydroxyl free group or to the 3 prime end. And we said that each of the DNA polymerase and RNA polymerase enzymes require a primase enzyme to build up a certain sequence that is called a primer, whose 3 prime end will be used to begin the process, either the process of replication in case of DNA polymerase or the process of transcription in case of RNA polymerase. And every time this primer or this sequence is an RNA sequence. This is very important. So why am I saying this? Because in case of DNA polymerase, we call it actually, or the best, the most accurate name is DNA primase. In RNA polymerase, we call it an RNA primase. This is a bit confusing because it gives you the impression that the DNA primase builds up a DNA sequence. No, because in both conditions, we build a single-stranded base, a single-stranded nitrogenous sequence. So it's definitely an RNA sequence with uracil instead of thymine. So the nomenclature comes from the name of the enzyme only. So both the DNA primase and the RNA primase do exactly the same function, but the nomenclature is different because the DNA primase belongs to the enzyme DNA polymerase, whereas the RNA primase belongs to the RNA polymerase. In the previous videos, we talked about the three types of RNA, and we said that messenger RNA is the delegate or the deputy of DNA that is capable of leaving the nucleus and going to the cytosol to be translated. We also talked about ribosomal RNA. I'm talking, by the way, about the videos that are questions and answers for revision of molecular DNA. I've recorded them, but I haven't sent them to you yet. You will not have to take notes of them, but they are full of questions and answers. So the worksheet will be, they would be very useful to you if you use them to answer the questions of the worksheet. Ribosomal RNA is also formed by transcription, but it is a structural RNA mainly because it enters in the formation of the ribosome, and you can check this in the videos. And it also has, it's not only structural, making the ribosome, because the ribosome consists of what? It consists of ribosomal RNA plus ribosomal polypeptides. So it's structural, but it also has an enzymatic function, and it's being said that the enzyme that peptidyl transferase and some of the enzymes that are used in translation are essentially ribosomal RNA. So we call it catalytic ribosomal RNA because it has an enzymatic function. Transfer RNA is just a type of RNA that carries the amino acid to the ribosome to help in translation, and we have explained this explicitly in parts 2 and 3. So this is the ribosomal RNA, where we assemble together the ribosomal RNA with the polypeptides to form the ribosome. In eukaryotes, you can have an 80S subunit and an 60S subunit. So you will also have a big part and a small part, a big ribosomal subunit and a small ribosomal subunit, but the number differs between prokaryotes and eukaryotes. Transfer RNA, as you can see, is clover shaped. It is essentially single-stranded like all the other RNA molecules, but it is because it has to take this shape, so there are areas that are double-stranded. If it happens that the nitrogenous bases that are opposite to each other are complementary to each other, then hydrogen bonds will form. If not, then like here in the loop, the hydrogen bonds will not form. We have three loops. We have the T loop, we have the D loop, and the anti-codon loop. What matters is the anti-codon loop, because if this is messenger RNA, so during the process of translation, here, for example, if we consider that codon, GAG, that corresponds to a particular amino acid here. And by the way, you don't have to memorize the names of the corresponding amino acids. Then if the transfer RNA has the corresponding anticodon, and by corresponding anticodon here, I mean the complementary anticodon, then CUC is complementary to GAG, then this is the specific anticodon. So the corresponding complementary specific anticodon means that the amino acid that is carried by that transfer RNA is in its proper place. So the transfer RNA via this anticodon codon hydrogen bonding will get the specific amino acid whose turn came on the in the polypeptide chain. What matters and what is more difficult is the stages of transcription. Just like you wrote in your notes about translation, which is the last process that we have: initiation, elongation and termination. Transcription also has initiation, elongation, and termination. So initiation starts by the promoter. So what is the promoter? The promoter is a sequence on the DNA molecule that will be transcribed where certain regulatory proteins will bind. So these regulatory proteins are called transcription factors, and when they bind, they help the enzyme RNA polymerase to transcribe RNA from DNA. And the promoter is upstream. Upstream means it is towards the 5' end of the coding strand. And we will see this on the diagram. So the promoter is where the promoter is a sequence on DNA. We can see this on the diagram. For example, here we have the coding strand of DNA, and here we have the 5' end. So this is upstream, and the other end 3' is downstream. So the promoter is this part. It's the first part of the coding strand of DNA. The first part of the coding strand of DNA. And the transcription factor, which is the specific protein that will bind here on the promoter that is present on DNA, will help the RNA polymerase know where to start. So we have the promoter, which is a sequence on DNA that is present upstream towards the 5' end. It's a long nucleotide sequence. It is non-coding, but it helps initiate transcription. So what is the promoter, or what are the main functions of the, what are the main functions of the promoter? The main function of the promoter is that it helps initiate transcription. It helps initiate transcription. How does it help initiation of transcription? By binding to transcription factors. So it binds to transcription factors. And when it binds to transcription factors, it tells the RNA polymerase where to start the process of transcription. Here we have what we call the transcription unit. So what is the transcription unit? The transcription unit is the gene that is to be transcribed, in addition to a promoter at the beginning, and in addition to a terminator at the end, together, of course, with the enzyme that is RNA polymerase. When we have a gene like this, it can code for a structural protein like actin, like myosin, like tubulin, or a functional protein like an enzyme, or even an RNA like transfer RNA or ribosomal RNA. Here we can see a more clear picture of the transcription factors. So here we have the enzyme RNA polymerase, and here we have the exons on DNA, the parts that will be transcribed into messenger RNA. And in between, we have the introns. You might have seen this in part 3. The introns are the part of a messenger RNA that will be excised or removed because they are non-coding. They do not code for a particular amino acid. And then here you can see the promoter, which is a long sequence of DNA that is present upstream towards the 5' end. And in the middle of the promoter, we have a sequence of nitrogenous base, thymine, adenine, which we call the Tata box. The Tata box is important because it binds this purple protein that is called transcription binding protein. The transcription binding protein is important because it is where the other transcription factors will bind. The presence of the transcription factors help orient the polymerase enzyme to where it should start the process of transcription. But ahead of the promoter, we have what is important to you is the enhancer region. The enhancer region is a shorter nucleotide sequence that is present. I repeat that we're talking about the DNA molecule. When I said here that the exons are the coding parts of RNA and the introns are the non-coding parts, it's the same, but the strand is DNA, meaning that the exons are the coding parts of DNA and the introns are the non-coding parts of DNA. So the enhancer is a shorter sequence of nucleotides, again, on the DNA molecule, on the gene in question. When I say the gene in question, I mean the gene that will be, in question, I mean the gene that will be translated. And of course, this strand of DNA that will be transcribed is the template strand of DNA. And the importance of the enhancer is to activate the process of transcription. So the promoter initiates, but the enhancer activates the process of transcription. And then afterwards, there is, of course, the process of elongation. We separate more of the two strands of the DNA molecule to expose more nitrogenous bases and to add more nitrogenous bases on messenger RNA. So the RNA polymerase can separate the two strands of the DNA apart, so it has the helicase activity or an activity similar to the helicase enzyme. And as we said before, the new nucleotides are being added to the 3' end of the growing strand as usual, but the only difference here is the complementarity of the bases. So C will pair with G, whereas A will pair with U instead of T. And we said again that the transcription unit is the sequence of nucleotides on the gene in question, the gene that is responsible for a particular trait and therefore for the production of a particular messenger RNA, plus all the sequences necessary for transcription. The sequences necessary for transcription, this is the coding sequence for RNA. So, but what are the sequences necessary for transcription? They are the promoter, the terminator, and we also have the enhancer that regulates the rate of transcription or how active and rapid the transcription is. We talked about the triplet codes, we talked about the codons. Sometimes a single gene can be transcribed by several RNA polymerases, meaning that, for example, if that is the gene, then an RNA polymerase will move onto the DNA molecule to build a new strand of DNA. What does this mean? This means that once an RNA polymerase enzyme leaves the promoter and moves towards the 3' end of the new strand or moves towards the 5' end of the DNA strand, a new polymerase, another polymerase will come and bind to the promoter, and so on. Every time the preceding RNA polymerase leaves its place on the promoter, as if the RNA polymerases were in a hurry. They look like a train or a series of cars that are one above the other, one next to the other or following the other. One enters through a gate, the other follows it in a sequence. And this happens whenever the cell is in a hurry to produce several polypeptides of the same time of the same type at the same time, because it's highly needed in the cell, like an enzyme molecule or a hormone molecule. At the end of the day, when this sequence is reached, termination of transcription occurs. Messenger RNA separates from the template DNA strand, and then, as we said before, messenger RNA has to be processed before leaving the nucleus. At the 5' end of DNA, we add a nucleotide, which we call a 5' cap. So this cap is nothing but a nucleotide that contains modified or a modified, actually, guanine nucleotide. And the importance of this modified guanine nucleotide is to indicate where the messenger RNA will bind to the ribosome in the cytosol. On the other hand, here we will have a polyadenine tail, which is a string of adenosine monophosphate, and adenosine monophosphate nucleotide. Why do we say monophosphate? Because every nucleotide contains only one phosphate. So when we say AAAA, this doesn't mean that we have adenine only, that we have a series of adenine bases, but actually they are nucleotides, meaning that there is the ribose sugar and there is the phosphate. And similarly, for the modified guanine nucleotide, it's actually not guanine, but guanosine nucleotide. The importance of the polyadenine tail is to protect the messenger RNA against being destroyed by the cytosolic enzymes. As we said before, there are areas of messenger RNA that are called introns and that do not code for a particular amino acid. We call them intervening sequences because, as you have seen in the previous diagram, they intervene between the exons. So if this is the DNA molecule, then the exons are the coding regions of DNA, exon 1, exon 2, and in between we have this intervening sequence. Why is it intervening? Because it comes between the two, and this intervening non-coding sequence is called the intron. We have what we call the small nuclear ribonucleoproteins. When I say ribonucleoprotein, I mean that they contain ribosomal RNA and, of course, protein. So their structure is similar to the structure of the ribosomes, but they are very small. Small nuclear ribonucleoproteins are called briefly SNRPs, because S small nuclear ribonuclear protein. So this is an acronym, and they are shortly called the SNRPs. Those small nuclear ribonuclear amino acids or nucleoproteins effectively are assembled to form what we call the spliceosomes. And what is the importance of the spliceosomes, it is to cut or remove the introns. And if we cut the introns, then the exons will be joined directly to each other, meaning that the messenger RNA, after having removed the introns, will become shorter and will become more effective because it does not contain redundant areas. So this is a kind of processing of messenger RNA because it prevents redundancy. In eukaryotes, we want the messenger RNA, when it goes to the cytosol, to be very effective. Every sequence on it should, or mostly every sequence on it, should correspond to a particular amino acid. So here you can see the SNRPs, the small nuclear ribonuclear proteins, how they bind together to form a spliceosome, and how this spliceosome binds with this pink area, and which is the intron, the intervening non-coding sequence. And this binding results in curving of this sequence that is non-coding in a preparatory step to remove it altogether from the molecule of messenger RNA and get a shorter, more effective processed mature RNA. Here we can see a very important diagram that comes frequently on the tests, which is that you have the template strand of DNA containing 3 exons that are colored blue and 2 introns that are colored red. And we have the process of transcription, of course, so we get a complementary strand of messenger RNA. We call it a pre-RNA or an immature RNA. Notice that introns are transcribed, meaning that we do not excise the introns from DNA. We wait until pre-messenger RNA or immature RNA is transcribed, and then we start capping, putting the 5' cap of RNA to protect it. So why haven't we added the polyadenine tail on the other side? Because this polyadenine tail is important only in the cytosol, because it protects messenger RNA against destruction by cytosolic enzymes. And here, this process occurs, is still in the nucleus. Messenger RNA haven't yet left the nucleus. So you cap first, and then you excise or you remove the introns. Excision here means cutting of the introns to remove them from the pre-messenger RNA. But splicing means binding. So excision here means to cut or cutting, and splicing means to attach. So after having removed intron A and intron B, you splice the exons together, and then you add the polyadenine tail just before leaving the nucleus through the nuclear pores to go to the ribosome, where the 5' cap will locate the messenger RNA and help it attach to the ribosome. And now you have a mature messenger RNA that has been processed. This occurs only in eukaryotes, because eukaryotes contain a nucleus. Eukaryotes have a nuclear membrane that separates the contents of the nucleus from the cytosol, but does not occur in prokaryotes. Thank you.